Okay. So we do have a midterm coming up on Tuesday, like I said. More likely than not in this class, because we just probably won't be able to get through each of the standards. We will likely end up taking a midterm that only is about half, it only covers about half of the Unit 5 standards. So what that means is that we'll have all of these red standards covered, all of the green standards, the 2 purple standards, 3 blue standards, and then 2 of these lighter purple or pink standards that we'll cover as well. The biology teachers and I are working to put together review videos, short review videos for each of these standards, in which we'll cover the most important topics. So you all will have an opportunity to review as you see necessary. Okay. So, quick question here. What are the subunits of DNA and what are their function? Can someone just tell me from A, B, C, or D? Thank you, Gabby. Yes. DNA is made up of nucleotides that store genetic information. Good. So DNA is a nucleic acid, and we'll talk more about that. But hopefully you all have taken this as a cue to take your notes out, because this is something that you certainly should have written at some point in Unit 1. And we're going to continue to talk about this in great detail in Unit 5. Unit 5 is all about let me go back to this first slide. It's DNA and protein synthesis, but what this really means is that this is the central dogma of molecular biology, the central dogma of biology, meaning it is the central, most important idea in a biology course, which is that the genetic information that is stored in DNA carries the instructions to make proteins, all right? And almost everything in our body is essentially a protein. We obviously have some lipids and some carbs thrown in, but most of what makes up our body is protein. So this central idea is that we can pass on the genetic information from generation to generation via DNA, and that DNA serves as the instruction manual to make the protein, which then makes up the organism. All right, so this is the most important idea in a biology course. For anything else, you can either type it in the chat or you can take yourself off of mute and say it out loud. Is there anything else that you remember about DNA? We've talked about it a good amount at this point. What else do we remember?
DNA is made out of nucleotides.
It's made out of
Nucleotides.
Nucleotides, good. Is there anything else we remember? Okay, nothing else. Well, again, hopefully you all you've taken this as a cue to take your notes out, because there should be quite a bit that you remember about notes. I guess you all are just being.
Someone put some information in the chat.
Oh, okay. Let me see.
I was gonna say they were made out of 3 parts.
Okay, good. Yes, Castro, a nucleotide is made out of 3 parts. That's good, and we're going to talk about those parts again. Kiki says every living thing has DNA. That's 100% correct. Kiki, thank you for adding that. And Gabby says that DNA can have mistakes. That's true as well. We have mutations that appear in DNA, and we know that the nucleotides can be in DNA A, T, G, C. Excellent. Okay, so we remember a lot. Good. And we're going to talk more about that today. Okay.
Oh, wait, it doesn't contain our genetic information?
Yes, ma'am, it does. Good. So I'm going to just uncover these boxes because we've already talked about it. But Deoxyribonucleic acid is a nucleic acid. The monomer is a nucleotide, and that is what Alicia told us. The structure, I don't think anyone said this, the structure of DNA is a double helix. So it looks like a ladder that has just kind of been twisted in a spiral form. The function of DNA is to store genetic information, and that's what Ayana just said. So good. If there's any information here that you need to write down, I would encourage you to feel free to do it. But I know you all at this point have seen this information before, so it's not super important. Well, it is important, but it's not important that you rewrite it if you're confident that you will remember it. Okay. Now, Castro said that a nucleotide is made up of 3 parts. Let me get my pointer here. We can see those 3 parts here. We've got a nitrogen base, which in this case is cytosine. That's why there's a C above it. C is one of those letters that Gabby told us about. Then we've got a 5-ring carbon sugar, a 5-carbon sugar. And then we have a phosphate group up here. So those are the 3 parts that Castro was referring to. Nitrogen base, 5-carbon sugar, phosphate group. The other thing we should point out in this small image over here is that the nitrogen bases are paired with one another. You can see that the C is a purple shape here. It gets paired with the green G. The A is this orange shape, and it gets paired with this beige or yellow T. We'll talk more about that. And here it is here. The nitrogen base, the deoxyribose sugar, 5-carbon sugar. And it's got 2 oxygens. You can't see them in this image, but that's why we call it, I'm sorry, it only has 1 oxygen. It has been deoxified. So there's an oxygen here, and there would be an oxygen here if this were ribose sugar, but this has deoxyribose. So we've removed this oxygen over here to make it deoxyribose sugar. And then we've got a phosphate group as well. All right. So the deoxyribose sugar and the phosphate group make up the backbone. So they look like the sides of the ladder. In this case, it's represented by this like shiny blue color. The nitrogen bases, the A, the T, the G, and the C, they make up the rungs of the ladder, which are the colorful things in the middle. Okay. So here it is again. We've got the phosphate backbone, which is made up of the deoxyribose sugar and the phosphate group. And then the nitrogen bases are in the middle. This is something that you should write down. And it's something that you'll talk more about in chemistry as well. But nitrogen bases in the middle are held together by weak hydrogen bonds. So these bonds, could be relatively easily broken, and they do get broken often when the DNA is replicated. So it's important that they are not strong bonds because we need to be able to separate these bonds for the purposes of replication pretty frequently.
I have a question.
Yes, ma'am.
So if they're weak, sorry if you hear my brothers in the background, but if there are weak hydrogen bonds, does that cause for easy mutations as well, or no?
No, not necessarily. That's a good question, though. But the purpose of the bonds really are in the middle here. And simply breaking these bonds just would separate one strand of DNA from the other, the complementary strand. And again, that's important because, as we'll see later, we need to be able to copy this DNA. We talked about that already. Remember, during interphase, during the cell cycle, interphase is made up of three parts: G1, S, and G2. It's that S phase in the middle during which the DNA is replicated. And so what we mean when we say DNA replication, we're just talking literally about copying the DNA. So in order to copy the DNA, and you all will see this in an activity that you'll do tomorrow, you literally just need to split it in the middle, and then there's going to be a special enzyme that comes in and it reads this DNA and it says, okay, well, this is a C, so that means I need to go find another G to put here. And this is a G, so that means that I need to go find another C to put here. So simply breaking the hydrogen bond doesn't necessarily make mutations more common or more likely. It just means it's just an important feature that the cell has in order to copy the DNA well. That was a good question. All right. And the nitrogen bases are complementary, meaning that they always pair up in this same way unless there is a mutation. So A should always be with T, T should always be with A, G should always be with C, and C should always be with G. So as you all are writing or thinking and processing through this slide, please keep in mind that the structure of DNA is always going to influence the function of the DNA. So what this means is if I change the structure, then I change the function. If I change how it is shaped or how it is organized, then I change the job that it's able to do. That's very, very important. Oh, oops. Oh, gosh, I always do that. Sorry. All right. So practice question. Remember, hopefully you all were thinking about all the ways that you can implement those test taking strategies that we talked about on Friday. Hopefully you tried some of them out yesterday, or you will try some of them out today as you complete your exam. But the question is, how does our DNA make us unique? How does our DNA make us unique? So keyword here obviously is DNA. We want to know how the DNA makes us unique. So if we do a brain dump about DNA, we know there are 4 nitrogen bases. We know that those nitrogen bases pair together, they're complementary. So as we look at these answer choices, does anything really speak to those facts? Someone typing in the chat. Oh, not yet. Any thoughts about what this answer might be?
I have an idea, but I'm not sure if it's right.
I couldn't hear that.
I said I have an idea, but I don't know if it's right.
Okay, well, share it with us and let's just see if it's not right, then we'll just talk about why it isn't right.
I thought it could either be A or C.
Okay, good. So let's look at the difference between these two. So Ayana says A or C. A says that the sequence of nucleotide bases determines the proteins you produce and the combination of traits you express. C says that the shape of each person's DNA is unique, making that individual show a unique combination of traits. So there are two things that I want to point out here that are really good that Ayana has identified. Number one, in both answer choices, Ayana has identified that the shape is important. Answer choice A talks about the sequence, which is essentially the shape. If I change the sequence, then I'm changing the shape. And answer choice C explicitly says the shape of each person's DNA. We also see the words unique. We also see, we see the word unique in answer choice C, which is a word that is used in the question. But the difference between these two answer choices really comes down to what is shaped differently. Is it the sequence of the nucleotides, or is it the overall DNA that is shaped differently? Well, let's go back a couple slides. This slide says that DNA is a double helix, and I think it said it here too. The structure of DNA is a double helix. So that's true for everybody. All living things have DNA. That's what Kiki told us. And all living things have DNA that is shaped in the form of a double helix. So when we go back, answer choice C says that the shape of each person's DNA is unique. That's not quite true. Everybody's DNA is shaped the same. It's all a double helix. So the correct answer is answer choice A. And I also like that Ayana essentially got it down to two choices here. And so by a process of elimination, she knew it wasn't B, she knew it wasn't D. And so in that case, she had a 50% chance of getting it correct. So that's a good test taking strategy too. Sometimes you're not quite sure, but you can really limit your options so that you're much more confident, even if you do have to guess, that you're more likely to get the right answer. Okay, so the difference ended up being that answer choice C talked about the shape of DNA being different. Not true. DNA is always going to be shaped in a double helix, but it's the sequence of the nucleotide bases that we want to focus on. How are they ordered? Is it ACGA, as we have here, or would it be CCGA, or would it be GCGA? So if we change the sequence, if we change the way that the four nitrogen bases are ordered, then we are ultimately going to have some impact on the traits that are expressed. Okay. So where do we find DNA in eukaryotic cells? Where do we find the DNA in a eukaryotic cell?
Nucleus.
In the nucleus, good. And what about in a prokaryotic cell?
Plasma.
Yeah, in the cytoplasm, good. Okay, so DNA is found in the form of a chromosome in a eukaryot's nucleus. So this is much more organized. But in a prokaryotic cell, it just swims around in the form of a plasmid. It does not, it's not surrounded by its own membrane. So that's the difference here. A nucleus is kind of like a cell within a cell. It has its own membrane. But in a prokaryotic cell, the DNA is not protected by its own membrane. It's just swimming around with everything else inside of the cell, primarily ribosomes. Okay. And as we kind of found out in that last question that Ayana answered, it's the order of the nitrogen bases that creates different proteins. And different proteins give you different traits. So it's the order of the bases that we need to be focused on. That's really important. Please make sure you write that down. It's the order of the bases that we need to be focused on.
Whose bit emoji is that? Is that one of the other teachers or something?
Yes, that's Miss Baxley. How observant of you. Are you saying that doesn't look like me?
No, that's the first thing that caught my eye, and it feels like familiar, like I've seen that before. Like I've
Yes.
seen a person that looks just like that at our school.
Ah, okay. So interestingly enough, we share quite a bit of DNA with species that we don't typically think are similar to humans. A mouse, for example, we share 92% of our genes with. So not even the DNA, not just the DNA, but the genes. 92% of the genes, which are specific sections of DNA that code for specific traits. So we share 92% of our genes with mice.
Is that why my brother chews everything?
That's probably more so due to his age. He's trying to explore the world. But even something like a plant, which obviously we would not really draw similarities to, we still have 18% of our genes in common with plants. It's quite a bit. Okay. So each human is 99.9% identical to any other human. In fact, we have very, very few genes that are unique to us. And again, a gene is just a section of DNA that codes for a specific trait.
I have a
Go ahead.
question.
Go ahead.
Sorry, my brother, he was trying to ask something too, but so traits like your parent, like I know my mom, she likes reading and she likes building stuff, and can that be like passed on, like, or you had to learn that?
Both. So this is one of the fundamental questions in science. It's this question of nature versus nurture. So it means nature is the perspective that all of our traits, even our personality, and even the things that we like to do, are decided by our genes. That's what the nature perspective represents. The nurture perspective represents the idea that your environment and your experiences contribute to who you become and what your preferences are and what your personality is. But in reality, you can't really decide that one is more important than the other. They always exist in some interplay. So some of your preferences and some of your mom's preferences are inheritable, meaning they can be passed on because they do exist in her genetic code. But in some cases, she has also supported that genetic code with experiences that have contributed to her preference and her desire to read and build things. So there will always be some type of interplay between genes and environment. You know, we can always think of professional athletes as being that interplay. You know, if Michael Jordan had grown up in Sao Paulo, Brazil, and had never been exposed to the game of basketball, then perhaps he would have become the greatest soccer player of all time. But it's also likely that he would just never have become he wouldn't have the athletic prowess that we know him to have had, right? So he has the genes that made him 6'6" and made him, extremely quick and muscular and had the ability to jump high. So that his genes made him that way, but also his work ethic, his life experiences contributed to his basketball abilities too. So in any case you can think of, there is going to be some amount of interplay between genes and environment. We can also think of it from a medical perspective. There is a gene called the Broca gene, which I'll send to the chat. BRCA. Supposed to be in all caps. So this Broca gene, of which there are two, I guess, one on chromosome 17, one on chromosome 13. This Broca gene essentially codes for a specific type of tissue in the breast. So both men and women obviously have breast tissue. Both men and women can get breast cancer, but it's obviously much more common in women. And so this gene will code for those cells that exist in the breast tissue. And if there's a mutation in these two genes, then a woman is much more likely to develop breast cancer than they would be if it weren't for that mutation. And so now science has advanced to the point where we can literally do genetic studies on women to determine their predisposition for breast cancer. How likely is it that they will develop breast cancer at some point in their life based on just looking at these genes and seeing if there is a mutation there? And that gives women the opportunity to say, okay, well, there's a 90% chance that I'm going to have breast cancer at some point in my life. I'm going to make the difficult but potentially life-saving decision to be preemptive and get a mastectomy before I develop breast cancer, meaning cut out that tissue. And Angelina Jolie is probably the most famous example of a person who's done this. But women do choose to do this sometimes because it removes the possibility that they could develop breast cancer, which could then obviously become deadly. So the gene, this mutation in this Broca gene makes it more likely that a woman will develop breast cancer. But there are plenty of women who do not have the mutation who still develop breast cancer. And they might develop it just by chance, or maybe they've developed it because they've been smokers for their entire life, or maybe they've developed it because they've been exposed to some amount of radiation. Sometimes when people are receiving radiation therapy for one type of cancer, they develop another type of cancer because the radiation is that amount of radiation is not healthy, especially if you go through kind of multiple rounds of radiation and you're dealing with multiple cancers at once. So the environment and the experiences that people have can also contribute to them having breast cancer, but sometimes it's a genetic disorder as well. Oops, I didn't mean to do that. All right, good question, Ayana. Ayana, you are one of the most inquisitive students I have this semester, and it's a great trait to have. All right, so I'm a little 7 minutes behind. We've got 28 minutes left in the class until 11:35, And you all have really just one assignment. Well, you have two assignments, the exit ticket, and there is an Amoeba Sister video with questions. And then you also have the opportunity to do test corrections and any missing work that you might have, any outstanding work. So that's how you should use your time today. I will be here on mute. Please let me know if you have any questions. Did someone else just join us?
I really think asking those questions is helpful.
Do I think it's helpful?
Mm-hmm.
Absolutely, yeah.
That makes me happy because my dad said to stop asking questions because it's unnecessary and stupid.
I will disagree with your father on that point. I love questions, and some of the greatest thinkers ask the most questions.
Yay, that makes me happy.
All right, folks. Yep. Bye-bye, Kiki. It is now 11:35, so you all are good to go. I will talk to you all tomorrow, and I hope you have a good rest of your Tuesday. Tomorrow is a early leave day.
That's